Welcome to another deep dive. Today we're going to be exploring some research that could really change how we think about HHV6B.
Yeah,
You know, it is that common childhood virus, but
Roseola, right.
exactly, roseola. But this study suggests it could be a lot more than just, a childhood illness,
Yeah,
especially for our HCT patients.
absolutely. And I think that's what makes this 2024 Hill study so interesting. It really takes a close look at HHV6B and its role in these lung infections that we see after HCT.
Yeah, and I think what I found fascinating was that they didn't just look at viral DNA. They looked at RNA transcripts in the lungs, too.
Yeah,
So we're talking about really trying to get a handle on whether this virus is just present or if it's actively, replicating and maybe even causing damage.
right. And they even went a step further and did whole blood RNA sequencing, which is pretty amazing.
Yeah, so they're really looking at the entire body's response to this virus, and that's something that I think is going to be super insightful.
I think so too.
So, for those of us who are dealing with HCT patients every day, lower respiratory tract disease is a big deal.
Huge.
It's a major cause of illness and death. And even with all the diagnostic tools we have, it can be really challenging to pinpoint the exact cause.
Yeah, think about IPS.
Oh, I know.
Idiopathic pneumonia syndromes. Like, we
Right.
do all these tests, and we still don't know what's causing it.
It's incredibly frustrating.
For everyone.
Yeah, absolutely. For us, for the patients. So I think this is where this study really comes in. They wanted to figure out,
Pin it down.
is HHD6B the culprit in some of these unexplained lung infections?
Yeah, and are we sure it's actually causing the problem and not just hanging out?
Right, exactly. So are we ready to get into the methods they used?
Let's do it.
Okay, because I think this is going to lay the foundation for really understanding the significance of their findings.
Okay, well, they did a prospective multi-center study. So, they followed a group of patients over time to see what happened, and they got information from different transplant centers, not just one.
So we're getting a broader perspective.
A real-world look at things.
Yeah.
And they looked at 116 adult patients who had gotten allogeneic HCTs. And they developed lower respiratory tract disease, LRTD, within 120 days of their transplant.
Okay.
And then they did bronchoalveolar lavages, or BALs, on all of them,
Got it. So they're going right to the source.
right into the lungs.
And they were very thorough. I mean, they didn't just collect the BAL fluid. They also got blood samples from each patient at the same time.
Which is super important,
Yeah.
to compare what's going on in the lungs versus what's in the bloodstream.
And here's where it gets really interesting. They didn't just test for HHV6B DNA in those samples. They also looked for mRNA transcripts.
Right, because DNA just tells you the virus is there, but mRNA tells you it's actually doing something.
It's like, finding the blueprints versus actually seeing someone building the house, right?
Exactly.
One is potential, the other is action.
I like that.
And get this, they went even further. They did whole blood RNA sequencing on a subset of the patients.
Wow. So they're looking at the activity of all the genes in the blood.
Yeah, and they're getting a really complete picture of how the body is responding to, in this case, a potential HHV6B infection.
It's amazing how much information we can get from these kinds of tests now.
I know, it's incredible. And then they didn't stop there. They followed the patients for 60 days after the BAL to see how they did.
Ah, looking at clinical outcomes.
Right. They wanted to correlate the HHV6B activity with survival, respiratory failure, really see if this virus was impacting how these patients were doing.
Makes sense. So they really did their due diligence with this study. They set it up well to get some really interesting information.
I think that's going to make the results even more compelling. So you ready to move on to those?
Let's see what they found.
Okay, let's dive in.
So they found HHV6B DNA in the BAL fluid of 37% of the patients.
Seriously, that's a lot more than I would guessed.
Right.
Yeah, I can see why people are excited about this study.
Yeah, it definitely catches your eye. But remember, finding DNA doesn't always mean there's an active infection.
Right. It could be dormant, just hanging out, not causing any trouble.
Exactly. And that's where looking at those mRNA transcripts comes in.
Yeah, that's the smoking gun, right? If we find those transcripts, we know the virus is actively replicating, possibly doing some damage.
And they found HHV6B mRNA in almost half of the BALF samples that were positive for DNA.
So that's a decent chunk of those patients with detectable HHV6B. They also had evidence of active replication.
Yeah, it starts to make you think that HHV6B might be more than just a bystander in these lung infections.
Definitely. So then what about those viral load thresholds we were talking about?
Well, they found that HHV6B DNA levels of 2.3 and 2.8 log 10 copies per milliliter, that was in the BALF, they were really good at predicting whether you'd find mRNA.
So if a patient has a viral load above those thresholds, we can be pretty confident that HHV6B is actively replicating.
That's what it looks like.
This could be huge for us clinically. To be able to order a test and know if HHV6B is a real problem for a patient with LRTD.
And it gets even more interesting when you look at how the patients did. They found that if patients had those higher HHV6B DNA levels, the ones above that active infection threshold, they were much more likely to die within 60 days of their BAL.
Oh, wow. So it's not just that the virus is active, it's associated with worse outcomes.
Yeah. And they actually found a link specifically to respiratory failure as a cause of death, too. So in those patients with higher HHV6B DNA levels, they had a higher risk of dying from respiratory failure.
Okay, so we're seeing that it's present, it's active, and it seems to be causing problems.
Yeah.
But what about the body's response? What did that whole blood RNA sequencing tell us about how the immune system is dealing with this?
Now, this is where I think it gets really cool. So they found that patients who had active HHV6B infection, especially the ones who had been diagnosed with IPS, they had different patterns of gene expression.
So the body is reacting differently when it's fighting off active HHV6B compared to other types of lung infections.
It seems that way.
Interesting. What did they see specifically?
Well, they saw strong activation of interferon signaling pathways, which is really interesting.
So is that something that we typically see with viral infections? Because interferons are part of our antiviral defenses, right?
Yeah, exactly. So it's almost like the body is recognizing HHV6B and it's saying, hey, we've got to fight this thing off.
And that makes it seem less likely that HHV6B is just an innocent bystander.
Right. The body is reacting to it like it's a real threat, like a primary cause of the problem. It's like finding the virus's fingerprints at the crime scene.
This is amazing. This could really change how we think about HHV6B and how we approach lung infections in our HCT patients.
It definitely has that potential.
But before we get too excited, we have to talk about the limitations.
Of course, every study has limitations. We have to keep that in mind.
Absolutely. So what were some of the main ones here? Yeah, no study is perfect. We always have to look at those limitations.
Right.
So what were some of the key ones here that we need to keep in mind?
Well, one of the big ones is that this was an observational study. It shows a link between HHV6B and these lung infections.
Right.
But we can't definitively say that HHV6B directly caused the infections or the increased mortality.
So there could be other things going on, other factors that they didn't measure that might be contributing to what they saw.
Exactly. And we also have to think about sample size. 116 patients is good for this kind of study,
Yeah.
but it's still a relatively small group, especially when you start breaking it down for different analyses.
Yeah. And I'm thinking specifically about that whole blood RNA sequencing data. I imagine the number of patients in that group was even smaller.
You're right. That analysis was only done on a subset of the participants. And while it's interesting, we really need larger studies to confirm those gene expression patterns.
So we need bigger studies, more patients, and ideally some controlled trials to really see if this is cause and effect.
That's the next step, for sure.
Okay, so more research is definitely needed. But this study is still a big deal, right? It's opened up a lot of new questions.
Oh, absolutely. It's a real game changer. It makes us rethink how we view HHV6B.
Yeah, and how we manage lung infections in our HCT patients.
Exactly. Now we need to figure out the best ways to diagnose and manage HHV6B reactivation in these patients and think about potential treatments.
Right, because how do we even treat HHV6B reactivation in these patients? Are there antivirals that work?
That's the million-dollar question. And right now, we don't have a clear answer. There are some antivirals that seem to work against HHV6 in the lab.
Okay.
But we need more data from clinical trials, studies in actual patients to be sure they're safe and effective in this setting.
So that's another area for future research. Do these antivirals really work for HCT patients with lung complications?
Definitely. And maybe even prophylactic therapy. If we can figure out which patients are at high risk, could we prevent these complications?
That's fascinating. So many new avenues to explore.
It's really exciting. This one study has just opened up a whole new world of research.
I agree. It's like a Pandora's box of questions, but good questions, important ones. We might be able to really improve outcomes for our patients because of this.
I think so, too.
Well, this has been a fantastic deep dive. We started with this common childhood virus, and now it's a potentially serious problem for some of our sickest patients.
And there's still so much more to learn.
Exactly. So much more to discover. Thank you for taking this deep dive with us, and keep those brains buzzing.